Hello, everyone. It's my great pleasure to welcome you to Global IMMUNOTALK 2023. I'm Kazuyo Moro from Osaka University and RIKEN. On behalf of the organizer of Global IMMUNOTALK, I'm very honored and delighted to welcome Sidonia Fagarasan from Japan as our speaker today. Many people know her outstanding study in mucosal immunology, but I really respect her for creating a new science by bringing immunology into metabolic and brain science field. So she holds MD degree from University of Medicine and Pharmacy, Romania, and PhD degree from Kyoto University, Japan. She's currently a team leader at RIKEN Center for Integrative Medicine Science, Medical Science, and also a professor at Center for Cancer Immunology and Immunobiology, Kyoto University, Japan. Her research activity includes impact of immune system on diversity, structure, and resilience of gut microbiota, and an extended group of activation, immunometabolism, and tumor immunology, and systemic immune responses impacting the brain biochemistry. She has a lot of co-authored more than 50 research papers in international peer-reviewed journals, and many of which are high-impact journals such as Science, Immunity, and Nature and Cell. So she has won many awards, including the first Kobayashi Prize in 2019, and her research is always highly regarded. So Sidonia, could you please turn on your camera for us? Okay. So first of all, thank you very much for joining the Global IMMUNOTALK. We're really looking forward to listening to your talk today.
Thank you, Kazuyo, and thank you for the introduction. I was a bit ashamed of your very nice talking about.
Oh, really?
I appreciate, and I'm delighted to talk today about our past and current work. Yes.
Okay, thank you very much. So before starting, I would like to know a little bit about you more, and want to make one question. And the question we love to know from you is, which are the most important gaps in knowledge that need to be addressed in the field of immunology?
Well, this is a difficult question, right?
Yeah.
So, we wish to have an answer for this. But I think immunology so far did extremely well in, starting from zero, essentially, and characterizing many subsets of cells and finding their function and fine-tuning, actually, the immune system even to fight cancer. This is an amazing progress. So yes, immunology did very well, but I think we have to even further push to understand the immune system, because we call it immune system. And I think we understand immune cells or immune populations, but still we are kind of not understanding the immune system itself and how it, how it's regulating and how the B cell and T cell really establish, the exact population or CD4, CD8 and so. So one thing is to understand the system, and then to understand how this immune system interact with other major system of the body, like major physiological system of the body. And we are making effort into this in understanding the connection between the immune system and the brain, but of course a lot of studies needed to understand the relation with cardiovascular or osteomuscular or gastrointestinal for that matter. And even though I worked for 20 years in with microbiota and immune system, I still don't understand the relationship really. So this is one thing, how the immune system influence all these organs essentially, and then it's also the opposite, how these organs influence the immune system function, because we all get the idea that when we are stressed, our immune system is not functioning. Therefore, the brain immune system connection is not only one direction, but it's also the other direction. So it's a lot to understand about this bidirectional way of interacting with all other organs. And I was trying to find a word of how we define this. And I was thinking for a long time at systems immunology, but maybe this is a little bit more confusing. And I would like to call immunosomatics. So the gap which we need to cover is this immunosomatics.
Very cool. But I sometimes really surprised how well you bring immunology into other fields, because we know about immunology, but we don't know about other side. So how can you do this? You need to learn many things from this, like a tissue matter or like a blah blah. How can you do that?
So first of all, it's to be humble and to admit that you don't know, and then is to left your comfort zone and to bring yourself into the unknown and to have the courage to explore that. And I'm really enjoying this, the unknown, and I like to learn. And I'm recently learning a lot from the biochemistry field. So I really enjoy reading the old papers, 60s, 70s, 80s. I love those papers, much more than I love the papers published these days. So they are fantastic, solid, short, concise, and very informative, I found.
Wow, enjoying science is, I think, the biggest thing for us.
Yes.
Okay, so let's move to your talk. Could you
Okay.
please share your slide for us? And if you're ready, you, yeah.
Is it okay?
Yes, fantastic. We can see and hear. So could you please start your presentation?
Okay, thank you.
Yeah.
So I will talk a little bit of this biochemical dialogue between the major physiological systems mediated by the immune cells. And as I mentioned, for more than 20 years, we are interested to study, we are studying the interaction between the cells, the immune cells, and the microbes. And we try to put this interaction in the context of the system. And we started with the gastrointestinal system. So, but you may ask why I was interested in this interaction. And this actually stemmed from my training. Like Kazuyo mentioned, I am an MD. I graduated University of Medicine and Pharmacy Iuliu Hațieganu in Cluj-Napoca. And actually, I was trained mostly in gastroenterology by Professor Ștefan Popescu, who was the last disciple of the Professor Hațieganu, who established the medical school, and the strength was internal medicine and gastroenterology. And what I learned in this med school was that medicine is art, and that we should be treating patients, not diseases. And I think these are highly relevant in these days. And then after gastroenterology, I moved to microbiology, and I became assistant professor in microbiology department. And of course, I was teaching microbiology and immunology to students. And I want to say that the department I worked was established by another it's a great personality, Victor Babesh, who was considered one of the founder of modern microbiology. And maybe many of you may know about Babesh-Negri bodies in the brain, which serve as a diagnostic of rabies, and also about the disease babesiosis, name to celebrate him. So I had this background in gastroenterology and microbiology from a very solid university. And so I was always interested in linking microbiology with immune system and diseases. And at a meeting, I heard a talk by Professor Honjo, later published in JXMed, mentioning about how the bacteria and different facility in mice elicited autoimmune diseases. So I got very much interested in this kind of work. And so I decided to move from Cluj, Romania, to Japan. And I arrived in Japan with a Mobushor scholarship for 2 years, and then my studies continue. And since then, I'm fascinated by this field of immunology and microbiology, and more recently, biochemistry. So I just want to start a little bit with the background of why the gut and how the gut developed. And I must say that this probably gut immune system started to develop when the big fish started to eat small bony fish and got gut injuries, which led to recruitment of some cells into the intestinal villi in the lamina propria. And this form aggregates and so recruitment of T cell and B cells. And what we are looking essentially is a system which evolved probably over 400 million years. And in mammals, the system is very complex because in fish, we don't have organized lymphoid structures, while in mammals, we do see that besides the intestinal villi, we have organized follicular structures. Some are containing one single follicle, agglomeration of B cell and T cell, and we call this isolated lymphoid follicles. But other structures contain multiple follicles, and these are called Peyer's patches after the name of their discoverer. So it is generally considered that these organized follicular structures are the inducive sites for activation and generation of antibody responses in the gut, while the intestinal villi contain mostly effector cells like T cell and IgA plasma cells. So yes, I mentioned IgA plasma cells, and because this is a common theme from fish to mammals. So the IgA production in the gut. You all know that in the systemic and peripheral immune system, we have IgG responses. But in the gut, the typical response is an IgA. And essentially, this is preserved from fish to mouse to human. In fish, we have IgT, teleost, which is the equivalent of IgA in mammals. And this is from an old paper showing in the lamina propria of a fish, some plasma cells, some IgM and IgA, IgT plasma cells secreting IgT, which lands on the surface of the epithelial cells. And this is a picture from the mouse intestine showing you many plasma cells, IgA plasma cells sitting in the lamina propria. So, and these plasma cells secrete IgA, and somehow these IgA are participating in coating bacterial communities existing in the gut, like shown here, the IgA on the surface of a bacteria. So IgA is a very interesting molecule. And I must say that we produce and secrete a large amount. So in human, it's like 3 or 4 grams every day of IgE production and secretion into the intestine. So this is another picture in which I emphasize essentially the plasma cells in the lamina propria, that you can observe the IgA transported across the epithelial cell, secreted into the intestine where it binds mucus, but more importantly, it binds bacteria residing in the intestine. So my very first question when I arrived in Japan was, what is the role of IgA? And in the textbook, what I was reading was that the IgA is related to elimination of the pathogen, because IgA is secreted into the lumen and it's inhibiting microbial adherence to the body surfaces, or neutralizing the toxins and viruses, or the activity of microbial enzymes. But so everything essentially was related to the defense against pathogenic bacteria or viruses. So I thought that it's a bit strange that we consume so much energy to produce this high amount of IgA daily, just to have it for prevention of a pathogen. So we embark in studies answering what is really the IgA role in the homeostatic condition in every day, for example. And so at that time, actually, I had the opportunity to study, to do the studies on IgA deficient mice. And because at the time I arrived in Japan, Professor Honjo was just discovering the AID, now the very famous molecule, activation-induced cytidine deaminase, which is expressed by B cell in these follicles in the Peyer's patch, the B cell, activated B cell organized in germinal center. And so, as you know, that IgE deficiency in mouse and human was characterized by the absence, is characterized by the absence of IgG or IgE or IgA. So mice and human deficient in AID have only IgM in the serum and in the gut. And of course, because AID is essential for class switch recombination and also for diversification of the repertoire by somatic hypermutation. So we started to study this, the gut in AID deficient mice, which had only IgM in the gut, but not IgA. And to my surprise, we found that in the absence of IgA, the AID deficient mouse had an enormous expansion of anaerobic bacteria in the small intestine, not much differences in the colon. And this was very striking. And recently, we begin to appreciate that actually the expansion of anaerobic bacteria in a wrong place, namely in the small intestine, now it's called small intestinal bacterial overgrowth, short SIBO. And there is the thinking that this SIBO is associated with chronic fatigue syndrome or with fibromyalgia or Parkinson and various other autoimmune diseases. So very important. And I want to say one thing, that the expansion of anaerobic bacteria in the small intestine cause a severe activation of the gut immune system. And this is just a photo of the intestine in a normal wild-type mouse. You appreciate this is a Peyer's patch, a protruding follicular structure. But in the AID deficient mice, we see these Peyer's patches as very much enlarged. And on top of that, we observe this tumor-like structure all along the small intestine. And these are nothing but hypertrophic isolated lymphoid follicles. And another message I want to give you is that this activation happens not only in the gut. It's not restricted to the gut immune system only, but is inducing activation of the whole body lymphoid organs. For example, you can look at the spleen. It's incredible hypertrophic, and this spleen has activation structures with germinal center, pretty much like you will see after a very, very severe infection or after immunization. So what we learned in the last 20 years, I think I can summarize like this, that in vertebrates, it was with the advent of this adaptive immune system, with the advent of B cell and T cell and molecule like AID, IgA, FOXP3 or PD-1, that allow the settling in the intestine of bacteria, diverse and balanced bacterial communities in the gut. And this kind of bacterial structures, communities, allow comfort us the genomic and metabolic functions which we don't have in our genome. And so generated these symbiotic relationships, which lead to the development of maturation of the immune system and to the phenomenon of oral tolerance, and in general of what we call health. And I think we also need to understand that this interaction between the immune cells and the bacterial communities in the gut are transmitted through biomolecules and signaling pathways to other systems, such as neuroendocrine or digestive and cardiovascular system. And so the immune system will essentially impact all these major physiological systems. And I mentioned at the beginning, we need to understand how these interactions and modulation happen so that we can manipulate in disease conditions. So recently we are focusing more to understand this metabolic interaction between the host and microbiota, and to integrate the gut, the endocrine, and the brain systems via the immune system. And so we thought to have to look how immune dysregulations would impact metabolome, and together will impact the blood biochemistry, and if this will have any effect on the brain function. And so to start, we started very simple, with a simple model. We choose PD-1 deficiency as a model of chronic mild activation. So PD-1, I think probably the audience is familiar with this, because PD-1 is used as a immunotherapy in cancer, and this was a game changer in cancer therapy. So what is PD-1? Is an inhibitory immune receptor expressed on the activated T cell. It has two ligands, PD ligand 1 and 2, expressed on antigen-presenting cells, and the engagement of PD-1 by its ligand is sending some negative signal to the T cell. So essentially function as a brake, leading to decreased activation or effector function of T cell. So the PD-1 expression, as you can imagine, is very important, because too much of the brake, too much PD-1 expression would be would mean immunosuppression, so which is bad for cancer or chronic infection. While the absence of PD-1 or PD-1 blockade can activate the immune system, but also can lead to an over-response, which we can see in patients treated, in cancer patients treated with PD-1 blocking antibody, leading to chronic inflammation and autoimmunity. So our question was whether the immune activation by the absence of a single immune inhibitory receptor can be read systemically, and if yes, how that will be. So just to summarize, we see that PD-1 deficient mice have a very complex phenotype. Of course, it was known by the work of Honjo and colleagues for a long time that the absence of PD-1 leads to expansion of T cell with pro-inflammatory properties. We also found that the PD-1 lack in T cell leads to expansion of T cell in the gut, particularly expansion of T follicular helper cells with pro-inflammatory properties, which will impact the whole biology and the dynamic of germinal center, and so will impact the production and the selection of IgA in those germinal center, and will ultimately impact the bacterial coating and what bacteria these mice will keep in their intestine. And more recently, we found that actually this activation can be read in the blood because we see a metabolite shift in the blood, which also impact the brain biochemistry, leading to behavioral changes. And I will mention just a little bit about this work. So we started to, we asked initially whether the PD-1 deficiency, activation induced by the PD-1 deficiency can be read in the biochemistry of the blood. And the answer is yes. When compare the small metabolites present in the wild type and the PD-1, we observe a significant decrease of many metabolites and increase of others compared to wild type. And among the most significant pathways affected were something related to mitochondria, but also with the tryptophan metabolism. And I was very interested in tryptophan because you may all know this is the precursor of serotonin, an important neurotransmitter. So in fact, when we look at the serum of PD-1 deficient mice, we see that most of the proteinogenic amino acids, not only tryptophan, but almost all other amino acids were decreased compared with wild type animals. And what I found interesting was that what we know, tryptophan, tyrosine, and phenylalanine, what we know as aromatic amino acids, were the first to respond to this activation. So you appreciate a significant decrease already by 2 months of age in these PD-1 deficient mice compared to wild type mice in these amino acids, which are very important for, I will later show, for neurotransmitters in the brain, but also for the regulation of the immune cells. So the front runner of amino changes by PD-1 deficiency are tryptophan, tyrosine, phenylalanine. And what I was surprised was that this happened not only in chronic activation of the immune system, but also when we activated specifically just very short time the immune system in the case of cancer immunotherapy. So for example, we injected tumor into a wild type animal, and we just injected 3 times anti-PD-1 antibody. And 2 weeks after, we look at the serum of these mice, and very surprising was to find that most of the amino acid, pretty much like in PD-1, were decreased by this PD-1 blockade. Suggesting perhaps that in human undergoing PD-1 blockade therapy, these changes in aminomes happen as well. And so then we ask, is this T cell dependent really? And yes, we confirm that in mice which don't have T cells and also deficient in PD-1, we don't observe differences. And so then we ask why these changes happen and what could cause, how T cell could cause systemic metabolome changes. I mean, this was really something which I never expected. So we try to look at maybe the liver, because in my school I was learned, I know that the liver is the kitchen of, and everything you read in the blood more or mostly comes from the liver. Or we thought that maybe there is some impairment in the gut absorption of amino acids because of some level of inflammation and disruption in absorptive capacity in those conditions. Or maybe we thought that simply the enlargement of these lymphoid tissues, like lymph node and spleen, will essentially trap these amino acids, which will be consumed by the activated immune cell to proliferate, to differentiate, to generate effector cells. And so we did the metabolome studies in all these organs, and what we found was that in the intestine, we did not see much differences in the metabolomes. In the liver, there were some, but where we did see metabolome differences at the tissue level were particularly lymph nodes, and some to some degree in the spleen. So these are the places where the T cell are expanding in the PD-1. It's very strange that the PD-1 deficient phenotype is mostly related to peripheral lymph node, so enlargement. And this is just to show that in the gut, there was not much difference between the wild type and the PD-1 related to these amino acids. But what we saw in the spleen, and particularly in the lymph node, was the opposite of what we saw in the serum. Namely, there was an increase in most of the amino acids in the lymph node or spleen. And again, this is the places where T cells expand. And just to show you how different is a lymph node in a wild type or a PD-1 deficient mouse, you appreciate that there is a huge, huge expansion of T cell in red, this red area, in the PD-1 deficient mouse. So this is not immunized, this is just, I think, 2 or 3 months age PD-1 deficient mouse. And so we visualize also this tryptophan, for example. I was curious if we can see the differences in the tryptophan level. And we did this in situ visualization of metabolites. And this is tryptophan. You appreciate that there is much more tryptophan trapped in the lymph node of PD-1 compared to wild type. And so I think we got the answer to this question of what causes the serum amino acid depletion, that it's induced in the PD-1 absence or even a short blockade of PD-1. It's T cell dependent, and it's a reflection of peripheral immune activation. And so, surprise, surprise to me, naive as I am, like T cell are orchestrating the systemic metabolome. And so we thought, okay, so we see the changes in the serum, in the blood, but what would be the consequences beyond the immune system? And here I come back to the point that tyrosine and tryptophans are the precursor amino acid for very important neurotransmitters in the brain. Dopamine synthesized from serotonin, so dopamine and norepinephrine, and serotonin derived from tryptophan. And so we asked how these amino acids and neurotransmitters will be in the brain. And it turned out that this was not a very easy experiment to do, and I was not aware how difficult it is to measure the metabolite in the brain. nonetheless, we could see when we look at the metabolome of the brain in wild type and PD-1, a reduction in tryptophan and tyrosine compared to wild type. But you know, brain is a very full of enzyme, easily degraded. So we were not satisfied with these minor changes in the amino acid levels. Therefore, we did a different approach in which we look in vivo dialysis. We did in vivo dialysis, measuring constantly the level of tryptophan and tyrosine in the brain of PD-1 and wild type. And here you appreciate much more than when you kill the mouse, that the tryptophan and tyrosine levels were very, very low and also flat, because we see this circadian rhythm regulation of the level of tryptophan and tyrosine, and these were not seen in the PD-1 deficiency. So this was very convincing that indeed, when these amino acids are trapped and used by immune cells in the peripheral lymph node, there is less reaching, passing through the brain-blood barrier into the brain. And so as a result of this reduction of precursor amino acid, we see also decrease in serotonin. Also by in vivo dialysis, you appreciate that when once you push the release of serotonin, it's much more released in the brain of PD-1 compared to wild type, and also dopamine and norepinephrine were significantly reduced. And I further wanted to see these neurotransmitters, and we had a wonderful collaboration with Yuki Sugiura. At that time was in Keio University, and he challenged and he really set up to visualize these neurotransmitters in the brain. I was not aware of any such studies before, and he did it. And I'm showing you here that yes, we could visualize serotonin in this place, which is called Raphen nuclei, which is significantly reduced, the serotonin intensity, the peak in the PD-1 deficiency. Also dopamine in the striatum, and norepinephrine in the locus coeruleus. So clearly we were convinced that this immune activation led to reduced neurotransmitters in the brain. And we know that these neurotransmitters are important for emotional responses. And so what this could mean in terms of mouse behavior. So we embark in another unknown and very challenging field of behavioral analysis. And we learn every single way to measure the behavior of mice. And we did essentially many tests, but what we found was that PD-1 deficient mouse were lazy. They were not moving very much. And they also had increased anxiety and also an increased fear responses compared to wild type animal. And just to give you a flavor of what the behavior means in mouse, for example, one test to measure anxiety is what we call elevated plus maze. So this is an elevated platform having two closed arms and two open arms. And one puts mouse in the middle of the platform. And of course, rodents prefer dark and walls. So the mouse will initially move between these two closed arms. And after a while, sensing that nothing bad happened, they will venture into the open arms of this elevated maze. And we can record this in, for example, the movement of the mice for 10 minutes. And this is a typical tracing of the mouse movement. You see a lot of traveling between these closed arms and then venturing into the open arms. But what we saw with PD-1 was totally different. They move much less, and mostly the movement was between the closed arms, and seldom the PD-1 deficient mice venture into the open arms of these elevated mates. So we can measure, we can quantify, and this is a sign clearly of increased anxiety of PD-1 mouse to explore, to go into the open field. And then we asked, okay, so the tryptophan is reduced in the brain. Can we supply by diet supplementation tryptophan? And if so, can we change the biochemistry in the brain and the behavior? And this is what we did. So we put the mice, both wild type and PD-1, on a tryptophan-enriched diet. So we supplemented just about 2% tryptophan in the diet. And we had also control groups in normal diet. And 1 month later, we look at the biochemistry in the serum, in the brain, and also we perform the behavior test. So what we observed was that, yes, the tryptophan diet increased and increased the level of tryptophan in the blood. And also what we noticed when we did a behavioral test was that now PD-1 on tryptophan diet started to move into the open arms. So the anxiety was decreased. But I must say that if we carefully look at the movement, we see that this movement is not quite kosher. So it's a very weird way of moving, in which moving and stacking and moving and stopping and something like that. But anyway, when we measure it, the travel to the open arm is clearly enhanced, and this correlated with the enhanced dopamine, increased dopamine levels in the brain, particularly in the raphe nuclei, where it should be. So supplementation of tryptophan by diet increased the serotonin levels in the brain, and we could rescue partly the behavioral changes, particularly the anxiety phenotype. So just to summarize what I talked until now, I said that in the absence of PD-1, there is an expansion of T cells, and that in the lymph node where this expansion is predominant, we see that there is a trapping of amino acids in this highly activated lymph node. And this comes as a cost, because then the amino acids essential are reduced in the blood, and then less it's going through the blood-brain barrier into the brain, and there is less neurotransmitter production, and this leads to behavioral changes in the animals. So the message I want to give you is that the immune system activation has a powerful impact on the blood and the brain metabolome. And of course, the connection between the immune system and the brain was known, because we know that neuroinflammation is related to these behavioral changes. But what I'm saying here is that you don't necessarily need neuroinflammation. We look into the brain and we look at the infiltration of T cell in the brain, and so we did not see much changes. So I guess what my message is that even if T cells are not in the brain, or there is not direct inflammation in the brain, still the brain senses the activation, the status of the immune system, because the activation of the immune system changes this biochemistry, and then the brain senses this and react and respond to this. And we all know that when we are sick, we don't move much. So perhaps this is one of the mechanisms of that. But I want to come back to the lymph node, because I was really fascinated by this complete biochemical change in the landscape of the metabolites in these huge activated lymph nodes in the PD-1 deficiency, where you observe that there are many metabolites increased compared to the wild type. And so we thought that perhaps during this activation of the immune cells, maybe they will generate some secreted immunometabolites, which will influence the function of other neighboring cells. And so we asked the question whether the secreted metabolites from the activated immune cells will serve as a communication language between immune cell, pretty much like we know, for example, B cell receptor or T cell receptor and the cytokines, ligand interactions. So we address this issue by a classical immunization protocol. So we immunize with complete Freund adjuvant a mouse in a foot bath. And 1 week later, we dissected the draining lymph node, which we call ipsilateral, and also we took the other contralateral resting lymph node to compare. We did this, you appreciate how much the draining lymph node of this immunization site is changed with a huge expansion of B cell and T cell, and we perform metabolomic studies. So, and as expected, the metabolome profile of activated lymph node, draining lymph node versus non-draining lymph node was quite different, essentially completely different. This is just to show you that in draining lymph node, again, many metabolites were significantly increased, some though were decreased. And among the most significant pathways changed by this immunization in the draining lymph node, it came glutamate, alanine aspartate and glutamate metabolism. And in fact, when we look at the most different metabolite, we spotted the 4-aminobutyric acid, which is actually known as GABA, the neurotransmitter GABA. And so we were really puzzled, and because we knew GABA just as a neurotransmitter, and in fact, it's an integral part of the mammalian central nervous system, and we knew about GABA as an inhibitory function, and this is related to the brain. But searching the literature and again going into unknown, we realize how ignorant we are, because in fact, GABA was found in plants and microbes. And in fact, GABA is in plant, serve as an adaptation to stress, for example. And so both bacteria and plants use this, what we later call neurotransmitter, for their energy source and for adaptation to stress, particularly drought or salinity or UV. So GABA is, and also in bacteria, for example, some bacteria make GABA, which help other bacteria to grow in a kind of symbiotic way. And this, for example, was found that Bacteroides fragilis supernatant contains GABA, and this helps other bacteria like Bacteroides, Parabacteroides, and E. coli to grow. So there is GABA modulating bacteria present in the gut. How about the body? So what we found was an old paper, 1978, from a Japanese group showing that GABA is present in other tissues in rats and rabbit other than brain. Of course, brain is very high, is the most abundant, essentially, neurotransmitters in the brain. But they found also in the liver and in the small intestine. And another study later identified GABA being produced by rat pancreatic beta cells. So what we decided was to look at the GABA in many different immune sites, as well as the brain. And I hope you appreciate that besides the brain, we can detect significant amount of GABA in lymph node, peripheral or mesenteric, in spleen, and much more than in liver, pancreas, or fat or muscle, where it was suspected to be. And again, we try to visualize GABA in the draining and non-draining lymph node. And I hope you appreciate the huge signal of GABA present, particularly in the B cell rich area, the B cell follicle, in this activated draining lymph node. So it's a signature, GABA is a signature of B cell activation, probably. And this is what we confirm because we sorted different cell population like CD4, CD8, or B cell or dendritic cell, macrophages, dendritic cells from the draining lymph node or contralateral non-draining lymph node. And indeed, we found GABA mostly produced by the B cells. And also, we further confirmed, because we were very nervous about this, the GABA being produced by immune cell, we wanted to make sure that this is not nerve in the lymph or in these tissues, and we are looking at the real stuff. Therefore, we did immunization also in the B cell deficient mouse, T cell deficient mouse, or both B and T cell deficiency, which is the rag animals. And you appreciate that in the absence of B cell or in the rag situation, we don't observe any GABA in the tissue. So clearly indicating that B cell lineage is a major GABA source in the lymph node. And then we look at many other tissues. We sorted the B cells from lymph node, spleen, bone marrow, Peyer's patches. And we also look at the IgA plasma cells from the gut, our old passion. And surprisingly, we found that most of the GABA is actually produced by the B cells, activated B cells, particularly IgA plasma cells from the gut lamina propria. And this is not a situation just present in the mouse, because we confirm the GABA production also in human, and this is a bad section of the tonsils from human. And I hope it's good enough to appreciate that GABA signal is mostly overlapping with the region where the IgA plasma cells and B cells are locating in this tonsil, while glutamine, which is the upstream, the precursor of GABA, amino acid precursor for GABA, is located essentially everywhere, because glutamine is required essentially by every cell to grow. And we also found in human, especially when we know that there is an activation of B cell, for example, in rheumatoid arthritis patients, that GABA was found to correlate with these autoantibodies titer in rheumatoid arthritis patients. So we kind of confidently say that increased GABA level is a reflection of activation of B cells and the presence also of IgA plasma cells. So because of the IgA plasma cell, it was known for some years, so in 2015, Michael Karin published I found a very nice paper showing that IgA plasma cells are in fact immunosuppressive because they are expressing high level of PD-ligand 1, which will engage PD-1 and block the response in this particular was prostatic cancer, non-responsive to treatment. And also because the IgA plasma cells secrete an anti-inflammatory cytokine like IL-10. So he proposed that there is a regulatory role of IgA plasma cells by IL-10 and PD-ligand 1 expression. And so that was one thing. And the other thing we knew from our studies was that B-cell deficient mouse have a very strong anti-tumor immunity, because if you implant a wild type or a B-cell deficient mouse with tumor, in the wild type, the tumor grows very fast. But in the absence of B-cell, tumor do not grow. And this happened both in the normal SPF, I mean colonized mice with all the bacteria, but it happened also in the germ-free animals, suggesting there is something intrinsic to B-cell, which is immunosuppressive. And so we thought about as PD-ligand 1, IL-10, and perhaps, GABA. And so this is just to show how different is a tumor in a wild type and in a MUMD in a B-cell deficient animal. So very powerful anti-tumor responses. And in fact, in the wild type, we can see some IgA plasma cells already infiltrating the tumor, interacting with the macrophages and also the T cell. So GABA we also found in some cancer cells, in some cancer tissues. For example, here it's a section from a renal carcinoma where we see infiltration of B cell and IgA plasma cell, and we stain for this not only for immune cells, but also we did visualization of metabolites like GABA and glutamine. And you appreciate again, pretty much like in the tonsil, the GABA signal is present, it's overlapping with the section where we see infiltration of IgA plasma cells and B cell. And in fact, we notice in the literature that there is a poor prognosis of renal cell cancer which have high infiltration of B cell. So obviously this kind of obvious question like the immunosuppressive property of B cell, GABA is involved or not. And so to answer this, we what we did was to implant a pellet with GABA in the B-cell deficient mouse. So this pellet will release constantly GABA in the mouse for about 23-30 days. And so we injected these mice with the tumor and we monitored the tumor growth. And what we found was that in the B-cell deficient mouse, now implanted with this GABA pellet, the tumor started to grow, suggesting that GABA partially inhibits anti-tumor responses in the B-cell deficient mouse. And indeed, in these mice, we look in the tumor microenvironment. We look at the effector CD8 T cell, which we know are very important to fight a tumor, to kill the tumor. So in the B-cell deficient mice, we see a lot of perforin and granzyme, typical molecules, which are decreased actually when GABA is present. And also the cytokine like TNF, interferon, very much increase in the B-cell deficient compared to wild type, but these are again decreased by the GABA pellet. So it seems that GABA suppressed the effector function of tumor infiltrating CD8 T cell. But it was not only that. It's much more complex always. And it was not only the T cell which were changed, but also the macrophages in the tumor microenvironment. So when we look at the macrophages in the wild type and in the B-cell deficient mouse, they were quite different in terms of transcriptomic. But in the B-cell deficient plus GABA pellet, they become much more like in a wild type. So there was a GABA shift in a transcriptome profile in mu-MT mice towards a wild type tumor-associated macrophages. And what was interesting was that among the transcripts which were upregulated in the absence of B cells and then downregulated by the GABA were those related with TNF signaling pathway or cytokine receptor. And there were some transcripts which were decreased in B cell deficient mouse, but increased by GABA. And these were related to cell cycle and with the oxidative phosphorylation. So, and this is just to show you how target gene activated by TNF, very, very highly upregulated in B cell deficient compared to wild type, and GABA really pushed them down. And the opposite happened. The OXPHOS genes downregulated in the B cell deficient mouse, but were upregulated by GABA pretty much towards the wild type phenotype. So I think we looked here at the induction of anti-inflammatory tolerogenic macrophages by the addition of GABA in this B cell deficient animal. But we wanted to make sure actually that is this really GABA produced by B cell, which is changing this situation in the mouse model of tumor we observe. And for this, we decided to do a knockout of the enzyme, which is responsible for GABA production in B cells. So we knocked down the glutamate decarboxylase, GAT67, specifically in B cells. And you appreciate that in these mice, the B cell produce very low level of GABA compared to the T cell level, basically. And heteromice were somewhere in between the wild type and the knockout animal. And so we implanted these mice with tumor, and this is the astonishing result I could not believe we got. So you appreciate an increased tumor growth in the wild type, an increased tumor growth in the hetero, GABA hetero, but a significant, powerful anti-tumor responses in mice which cannot make GABA by the B cell, but they can make GABA by other cell, okay? So this strongly indicated that at least in this time point in this model, the B cell specific GABA deficiency is enhancing anti-tumor responses. And I think this is very important. And of course, this associated with an increase in killer T cell capacity as we see more perforin, granzyme, and these inflammatory cytokines in the GABA deficient B cell. And so just to summarize what we did many other experiments, but just to summarize, what we think is that activated B cells or IgA plasma cells produce and actually secrete GABA. And somehow this GABA is conditioning monocytes to differentiate into anti-inflammatory macrophages, arginase positive, IL-10 positive, anti-inflammatory macrophages. And somehow this modulates the activity of killer T cells and inhibit, in this case, the activity of killer CD8 T cell, and the tumor can grow. And actually, I did not mention, but the GABA is working also directly on CD8 T cell by activating GABA A receptor and sending an inhibitory, actually resulting in an inhibition of T cell suppressive activity. So in the absence of the B cell, or the monocyte differentiate into inflammatory macrophages, and somehow this inflammatory macrophages modulating the function of killer T cells such that they produce more granzyme, interferon, and they are dealing much better with the tumor. Of course, there are so many questions that we need to address, and among the very obvious one is where this conditioning happens and how exactly, because the GABA is very complicated system, pretty much like in the brain. Like there are two types of receptors, receptor type A and ionotropic, and receptor B, which is a metallotropic receptor. And there are many transporters for GABA in the brain. Well, surprise, surprise, all these are preserved in the immune cells. And so we need to know how GABA is working. Is it working through the receptor A? Is it working through the receptor B? Is it working through the transporter? Is it extracellular or intracellular? If intracellular, how is that going? What is doing? Is like in bacteria or in the plant, is producing energy, it's binding protein, is modifying the function. Many questions of a neurotransmitter, which was originally designed not a neurotransmitter, I believe, but rather as a growth factor and as a regulator and of adaptation to stress. And yeah, immune activation is a stress. So, and sometimes I want to say, and I don't want to give a wrong impression that only B cells can make GABA. No, sometimes the cancer cell can themselves produce GABA. And this is one example. For example, in the prostate cancer, we could see, not in all, but in certain patients, that tumor cells produce themselves GABA. This was not infiltrating tumor, yet GABA was present. And actually, a paper published after our work was published identified that GABA produced by cancer cell in fact promote the growth of the cancer and also inhibit the CD8 killer T cell responses. So I think it's becoming very obvious that targeting GABA signaling is an option for the cancer treatment. So to summarize my talk, the later part, we identify the neurotransmitter GABA as a signaling molecules which is produced and secreted by B cell and plasma cells, and that this B cell derived GABA facilitate the differentiation of macrophages with anti-inflammatory properties, and that they are suppressing cytotoxic function of CD8 T cell and suppressing anti-tumor responses. So I would suggest that we should look pretty much like what's happening in the brain with a lot of neurotransmitters and receptors and their work. In fact, immune cells are also communicating and regulating by these neurotransmitters. And so there is a similarity between the brain and the immune system, which many people thought for a long time, but it was difficult to gauge. And I suggest that there is a common language of communication employed by immune cells and by the brain cells. And one example I gave today is the secreted immunometabolite GABA. And I would further suggest that the small metabolites actually have a great evolutionary potential as communicating molecules because they are synthesized and secreted with much more rapidly and using the fewer cellular resources than the classical, receptor, protein receptor signaling pathways we know in immunology. And so what I'm thinking about the immune system function, when I come to Japan and I knew just gastroenterology and microbiology and some immunology, I saw that the immune system is basically designed to protect against infection. And of course, we knew this as infection and immunity. So in the last 2 or so decades, we understood that the immune system is essential to establish and to maintain healthy bacterial communities in the gut. And this is what we call symbiosis. Although we don't understand what healthy community bacteria means, but I would say it's just the location. So the bacteria should be located where they are supposed to be, not in the small intestine, but rather in the large intestine. And there is a balance between the 2 major phyla, Firmicutes, Bacteroidetes. And this is what I call a healthy microbiota, roughly. And what I think we still need to understand, and I mentioned in the discussion with Kazuyo at the very beginning, we need to understand the role of the immune system in regulating the function of other major physiological systems of the body. And I would like to call this immunosomatics. And this was suggested to me instead of systems immunology. Immunosomatics was suggested to me by Antonio Coutinho, the former director of the Gulbenkian Institute, who said, well, this is what you should call this integration of every systems with the immune system. And I just want to mention that I could not do anything without these fantastic people, young and motivated, embarking in many experiments totally unknown, and as I said, getting out of the comfort zone, and of course great collaborators in Kyoto University, in former Kyoto University with Yuki Sugiura. We did all metabolome and proteomics. Fantastic Ibo Wu, who is now in Geneva. And yeah, I think we all appreciate these images of the galaxy clusters by the telescope, but we have to launch our telescope in our own body. It will be even more fascinating. And I think the journey just began. And I finish here, Kazuyo.
Thank you very much for your excellent talk, Sidonia, as always. And I understand very well why you got interested in the concept of immunosomatics and why such a comprehensive idea is required for the future of immunology. So I really appreciate your kind talk, and again, thank you for sharing the wonderful study with us. Thank you very much.
Thank you for giving me this opportunity.
Okay. So let me show one slide before closing to let you know how to make a question to Sidonia. Okay. So if you would like to ask Sidonia any question, please do it through the Twitter. And please search for account Global IMMUNOTALKS and find tweets that say ask question for Dr. Sidonia Fagarasan here. And reply to the Twitter with your question and mention hashtag Global Immune. So please note that due to the time difference, we are in Japan. There may be some time lag in Sidonia's reply. So and also, we usually introduce the next speaker, but since this talk is pre-recorded, the first speaker for the second half of the year has not been announced. So please check our website for next week's speaker. So thank you, Sidonia, again. And thank you. We would like to close today's seminar. Thank you very much.